Okay.
Okay, so my research study is entitled Evaluating the Impact of Banana Feeding on Honeybee Brood Rearing and Colony Health. That was a study that was done, a paper that was done by Katarina David, master beekeeper, on Cavendish bananas that increase brood rearing in Apis mellifera. And I wanted to do a peer review on that paper because there was no other study that was looking at that. So for the purpose of this presentation, I'm going to call Katie Katarina David. That's short for Katarina David. And for those of you who don't know me, my name is Nathalie B. I'm with Be Mindful. I'm a master beekeeper. I've been doing bees for about 15 years. And I have about 400 colonies, all treatment-free. And I specialize in natural horizontal beekeeping with tabar hives. I have 2 teaching apiaries, workshops, and apprenticeships. And I'm also the inventor of the Flex Hive that combines Langstroth, that adapts Langstroth to tabars. So as most of you know already, bees eat a 100% vegetarian diet, which is made mostly of pollen for sources of protein and growth and fat, and nectar for carbohydrates, fuel basically, and energy. Pollen, it can be diverse, and they help build bee bodies. That's what they're fed to the larvae. And the nectar is fed to any flying actor in the hive. So I'm going to go through some of those basics pretty fast because there's a lot to talk about. But what prompted the analysis by Katarina David is that bees sometimes are seen feeding on fruit juices. It just so happens that usually it's during a dearth when there's nothing else available. And instead of piercing the fruits, the bees are opportunistically leveraging fermenting broken fruit, not foot. I broke my foot this year. To drink the sugary juice from the exposed fruit flesh. So that's a kind of a backup, kind of a source of carbohydrates on all kinds of fruits. Everybody's seen the watermelon ones where the bees go crazy. So that's kind of what prompted Katarina to look at bananas and see if they're any good for feeding bees or as a supplementation. The reason is that they're made out of protein, fiber, starches, and sugars, including some of the bees' favorite ones like fructose and glucose. And there are similarities in some of the components of bananas with pollen in the macro and micronutrients that could be potentially fitting for a bee's diet. And so the evaluation is looking at if it's a good fit for bees. So the purpose of Katarina's research was really to investigate whether Cavendish bananas specifically offered any real nutrition or brood rearing benefits to bees. And the objective was to test whether banana feeding increased brood production. And she used packaged colonies for that in a period of 30 days. The goal was to determine whether bananas could serve as a practical, economical supplementation that could help strengthen colonies during key periods, especially dearths, because it's cheaper than sugar in some of those countries in Africa, for example. Maybe not in the US, but in Africa, that was the case. So her experimental design included 12 packages split into 2 groups, a control and a test, into 6 frame Langstroth nooks built out of plywood. And the banana fed test group and the control group were installed mid-May in Kenner, Oregon. It lasted about a month because at the end of the month, the colonies outgrew the boxes. So she had to stop. So she had 6 on one side for the control and 6 on the banana fed test in different areas of her yard. And what was noticeable right off the bat is that when she started her study, very quickly, the banana fed test group lost 2 of the 6 colonies, leaving her with 4. The control group, the one that was fed only sugar syrup, lost only 1 colony, but was left with 5. So both groups were fed sugar syrup, and the test group was fed bananas as well inside the hive. So she was left with 5 colonies control and 4 colonies in the test group. The control group, again, had 1-to-1 sugar syrup at the rate of about a quart a week. And the test group had the same thing plus ripe banana pulp and peel, up to one banana per week. If you can look at the bananas on the right side, she was going for fairly ripe, but not too dark. So her feeding setup was packages in those 6-frame Langstroth nooks with an extra super at the top where she could fit a box for the banana, the blue Walmart box, and a feeder, round top feeder, where she would put the sugar syrup. So it looked like this. And then she would weigh the colonies weekly and evaluate the brood surfaces weekly as well to see the variations. So her data set and results showed various pieces of information where she had the cause of the losses of the colonies out of the data was due to queen failure, both 1 in the control group and 2 in the test group, which is pretty high, by the way. So I wonder what caused those queen failures. The weight changes, the data she put together in bar charts, and you can see the left is a control group and the right is the test group with the bananas. So you could see that the control group weight changes seem to be higher in general than the ones with the banana on the right side. Same thing with the number of brood surfaces. It appears that is the opposite actually. The control group had fewer brood surfaces and the banana test had more brood surfaces according to Katerina. So what she found was the test group was lighter and gained 2% less weight, meaning the banana fed 1. And it also had an additional 2 frames of brood compared to the control group. And it also reared about 18% more brood according to her results. And since the bees ran out of space after 1 month with up to 12 brood surfaces, then she discontinued the study. So right off the bat, I noticed a couple of things. The test group had twice the number of queen failures. The sample size was not very statistically significant. That's a very low number of colonies because she was left with 4 on the test group. The study ended up after a month, which is rather short amount of time in the life cycle of a colony of bees. The amount of syrup consumed was not recorded, which is an issue when you're trying to eliminate confounding variables. And the brood surfaces were visually counted in a quarter of increment of a comb, of a frame, which is not very precise and is very subjective as well. There was no recording of the brood pattern and its health either. So some of the issues I took with that study. This is very, small, but I will walk you through that. Basically, she was using some resources based on studies that were done by a couple of people, namely Akinwande and Nepan, as well as Pandey in her own. So what her observation was that the Akinwande study, the first one, and I'm happy to share the slides with that, but mostly those I found were not very relevant because they were based on swarms that were caught. And so that kind of means the size of the colonies and the composition of the colonies might have varied quite a bit. That's a confounding variable. The other thing is that the brood surfaces were not counted, just the foragers and the weight of the harvested honey. So that is not very much a comparable study to what she's trying to accomplish. Nonetheless, the test colonies, the banana colonies, showed a 7.5% higher population and a 23% higher honey production when feeding banana puree syrup. That was not just bananas, that was bananas mashed up in sugar syrup. So also not comparable. The second study, Nepan, also had a different feeding protocol where they were using, for all the colonies, 500 milligrams, about a pound of pollen substitutes. That kind of is interfering with the purity of the study as well. And they were feeding banana syrup at the rate of 100 grams of banana pulp with 1 liter of water, and then filtered that and mixed in 200 milligrams of cane sugar and 20 milligrams of honey. So there's a lot of things going on in that study as well. Nonetheless, when they were fed only sugar syrup, that's the control, it appeared they increased the number of brood cells by over 50%, 53%, and the number of brood frame by 37%. Whereas when fed only the banana syrup, it was a lot lower, 16% and 17%. So actually, it disproves what Katerina is trying to do, even if we disregard the different protocols. And it was something fairly similar for the Pandey study, where there was cane sugar and honey mixed in with banana syrup. Now, again, her study showed an 18% increase in brood surfaces. My study, I will show you what the results were. I was expecting, I had somewhat of a bias. All researchers might have a little bit of a bias when they first get started, but I was expecting that it was not going to be the case, that it was not going to be increasing brood surfaces, that it was not going to make any difference. What I found out, though, was quite shocking. So here's the project purpose for my study. The background was Katie Katerina's paper promotes feeding bananas as a natural bird stimulant, yet the scientific evidence remains limited and conflicting. A lot of the studies she would use were not comparable and didn't relate to what she was trying to do. Given bees' nutritional and behavioral sensitivities, bees are a superorganism that has a lot of interactions going on. It's vital to verify her claims for responsible apiculture. So the objective is to really provide a peer review and to critically evaluate the scientific validity and the practical implications of her study so that we can decide if banana feeding is positively affecting brood production or not. The goal in the end is to assess her methodology, the biological plausibility of her conclusions, and the potential unintended consequences of that feeding protocol of feeding bananas. The ultimate goal is to enable beekeepers to make informed, evidence-based decisions that are grounded in bee biology and sound science, not just, assumptions. So the methodology and design that I used was as follows. We're going to talk through the experimental design, the materials that I used, the feeding and data collection setup, the control and the treatment feeding protocols, why I chose this design, and the key variables measured, as well as the data collection method, which should be fairly similar to Katerina's, right? The goal is to provide a peer review. And in the end, I will look at the results and offer some conclusions and some considerations. So let's look at the experimental design overview. Instead of using 6 colonies in the test and 6 colonies in the control, I used 15 of each. So I self-funded and bought 30 packages of Italian honeybees, same as she did, but more. And because I'm a top bar horizontal beekeeper, I put mine in horizontal top bar hives because I'm eliminating the plastic foundation potential interference, the confounders. And I wanted natural comb, and I was going to integrate that in my operation later anyway. I'm using my standardized plans, and both groups were separated into 15 and 15 on different area of the same yard. Like that's a 7-acre property at my home yard. So one group on one end and the other one on the other side. I started my study on the 3rd of April, and same thing. I compare apples to apples. I ended it after a month, which by that time, a lot of those 10 bar top bar nukes were running out of space also. And it was done in Driftwood, Texas. I'm my teaching apiary. So what I ended up looking at where Katerina had 6 and 6 initially, and she lost 2 and 1 on each side. She was left with 4 colonies for the test and 5 colonies for the control. And my test was with 15 on each side, and I lost 3 colonies to queen failure on the banana fed test, but none on the control, which already leads me to ask, why is it feeding bananas seem to have such a high rate of queen failure? There might be something going on there that needs further research to confirm. So the design overview Same as Katerina, the colonies were standardized for queenage because I bought packages, genetics, and population size before the trial onset. Those packages are great for that. The brood coverage, the brood surfaces, and the weight gain, as well as the consumption behavior in terms of sugar syrup and banana, were recorded weekly for all 4 weeks. And the data was analyzed for percentage changes in both brood coverage and colony weight. Those are my 2 measured variables. This is what a tabar nuke looks like. I don't have an actual picture of it. I have one in my car, but I didn't bring it out. And it's based on standardized tabar nuke plans. The plans are free, easy, do it yourself. You can download them and do it yourself. The nukes bodies were built with also 1-inch boards of they were 1 by 10, and they were a little over 16 inches long, and they were pine, right? And they each held 10 bars, same exact configuration for all the colonies. The bars, the top bars that were used, were built out of 2 by 2-inch thick wood, and they measured 20 inches long. That's the standard also for my operation. Rain covers were made out of marine plywood, and all the boxes, same as Katerina, were built on site using the same materials, pine, plans, and dimensions to ensure uniformity. And also, I will show you later the same configuration with the box on top and the feeders above, with the bars where I drilled a hole through the bars about 1 inch diameter, so that the bees could go up and get the food, the banana from the blue box, and the sugar syrup from the round top feeder. Why did I use those? In nature, bees will build horizontally and vertically, actually a lot more in horizontal configuration, it looks like. And this is why I use those. If you look at the entrance, the bees will put their brood nest near the entrance for ventilation and thermoregulation. And then they'll start building comb and put in the back the honey combs where the surplus food is stored. So what happens when they do that, they have a brood's nest by the entrance. They also have honeycombs in the back, and the bird's nest is usually, there's also a little bit of honey above the bird's nest, and at the front, there's 1 comb of honey by the entrance. And usually the bird's nest is surrounded by bee bread, which means same configuration every time. It makes it super easy to figure out where everything is. The inspection starts from the back, and as you pull a couple, 3 combs initially, then you can start traveling that opening until you reach the bird's nest. Then you don't have to disrupt the bees to inspect them. You could just leave the integrity of the bird's nest intact. So the top bar nuke functions the same way. And this graphic is not very good because I'm not very good at doing graphics, but it's the same thing, but in a smaller box, basically 10 bars instead of 32. And so they'll build the same way. And when they're full, we'll stop the study in this case. So again, these take very little resources, a few boards, and you can make a huge one. So they are double, and they cost very little per colony to build. In just 90 minutes, you can have a double hive. So my feeding setup pertaining to this research, again, I chose packages, 30 packages. Each went in a top bar nuke and got installed. And then this is not very pretty because I don't have a photo, but the medium box for an 8 frame was big enough to give space for both the little blue box from Walmart and the top feeder, the round top feeder. And on top of it, I had a cover to make sure that it was protected from the sun and the rain. And same thing, I used a scale to measure the weight and visual observations to measure the bird surfaces. So my feeding protocols, again, this is a peer review. So I use the same feeding protocols with a control group with sugar syrup and a test group that had sugar syrup and bananas on top of it. Why did I choose this design? Because I wanted a peer review. The reason I used the top bar nukes is because I wanted natural comb to minimize wax contamination and VOC's interference from plastic. All the equipment was new and identical for standardization. The boxes, the top bar nuke boxes, allowed for rapid brood expansion, just like with Katerina's Langstroths. And it was easy to measure the weight gain and observe and it was compatible with my operation. The control and the test were separated clearly on each side of the property to reduce experimental contamination, drifting, rubbing, things like that. And the much larger sample size, the N, was chosen to improve the statistical power of my analysis, right? You have a much better, much more confident results when you have a higher number of data. And so that it would absorb random variations as well. And it increases, in this case, the reliability of the analysis of variance for this study. So that was the reason why. The variables that are measured were the same as Katarina David's. The independent variables, the cause that we're testing, were the presence or the absence of banana feeding, right? It's a binary, yes or no banana. The dependent variables, the measured outcome, were the brood surfaces and the weight, right? And then the control variables, the constants, were everything else that I just discussed, the genetics, the colony format package, the queen age, the style, and so on and so forth. So basically, I use the same protocol as Katarina David did, okay? Trying to compare apples to apples. The data collection method. So when Katarina was evaluating her brood surfaces, she was counting it in a quarter increment of a brood frame. I counted it in an eighth of a brood surface, right? The weight was also collected in grams using the same scale for all of them after removing the feeders and banana remnants so that it would be just colony and box, and the box's weight was similar and accounted for. The brood area measured was again in 1/8 of a comb to increase granularity and make sure that it was more precise on both sides. And the collection dates and times were the same for all of them within a 2-hour window, because it takes a while to weigh and observe 30 colonies like that, actually 27 in the end. And the reason, again, for this data collection method was the reproducibility of the tests for the peer review and additional credibility on the study. The differences in finding because of this exact protocol was due to biological variations, right? Biological factors, not really data collection or protocols. The data collection was also weekly, so fixed intervals, so that it's kind of representative of the progression for both weight and brood surfaces. And because of the consistent timing and method of measurements, the results were controlled. So that's why I chose this. My role in this design, again, I was the funding source. I paid for all those packages and built all those boxes on my dime. I was the lead researcher, project manager, and principal investigator. I designed the methodology. I led the whole project. I was by myself, so it was not difficult to lead myself. I don't get to disagree with myself, I guess. And I was my own scientific oversight. So maybe I should have gotten somebody else to provide a little bit more scientific oversight, but I did it on my own, this whole project. So the field tech was, again, me. I was the one collecting the data, and I was also the one making sure the protocol was followed. So for the setup, for the feedings, and for the weighings, and for the brood surfaces measuring. So in the end, once I collected all that data, I was also the data analyst, and I'm also the presenter. So from A to Z, I'm the one who's doing it all. And I assembled the graphs and tables. I calculated and present it. Right now, I'm presenting the results. And I also opened up the discussion, and I'm in the process of writing a paper to publish that peer review publicly. It's not quite done, but right now we're going through the slides instead. So let's look at the data collected and the results that this peer review generated. The way I looked at the brood surfaces, and it's kind of what Katarina did as well, is I was counting the variations since the last visit, but also the overall variation from the beginning to the week where I was at. So at the end of the 4 weeks, that was from week 0 to week 4 since installation. Same thing for the colony weight between visits, but also overall changes in colony weight. So these are my results that I summarized in this table. You can see that the control group has 15 colonies, and that the test group is a little bit fewer rows because it has only 12. So each of those colonies is a week, 403, 410, 417, 423, and 430 at the end of the study. And each of those colonies was named 1 to 15 in the control group and 1 to 12 in the test one. So I calculated the weight increase for each of these weeks, and then the overall weight increase for the whole study, and then the weight increase percentage, and the number of brood surfaces for the entire time frame. So what this looks like in terms of weight variations, in terms of bar charts, this is the control group. The average growth was 46% for the weight, which is to be expected that they're going to grow. But the test group, the one that was fed bananas, was actually a little bit less weight gain, probably less honey or nectar. And the growth was 36% overall, average. So you can see with that line, the control group is going over the line when the test group is not quite reaching it. So overall, over the entire period, that means that the test group, the one that was fed bananas, gained 22% less weight than the control one that was fed only sugar syrup. So they didn't gain as much weight. Same thing with the line charts, same numbers, but just a different visualization so that, you know, some people are more line charts versus bar charts. And now let's look at the brood surfaces results. So we have same thing, control group on the left and test group on the right. And from week 1 to week 2 to week 3, and then in the end, we have a 5.8 brood surfaces, and then for the control group. So for the test group, the one that was fed bananas, I was very surprised to see that actually there were fewer brood surfaces. Instead of 5.8 at the end of the last week, it was 4 brood surfaces. In effect, that was almost 2 combs fewer than in the sugar syrup group, and a minus 31% brood surfaces increase. I was shocked. Overall, the average went from 4.35 for the sugar and 2.92 for the bananas, which means that the overall across the entire study for the entire month was minus 33%, almost 1.5 combs of brood surfaces fewer. Remember, I had a bias. I thought it was not going to make a difference. I was shocked to find out it actually decreased the brood surfaces by quite a bit. So this is another way to look at it, the bar charts. And then this is the brood surfaces in bar charts as well. And then this is the line charts. So I try to replicate Katarina's protocols as closely as I could, her objectives, her method. But I think that to improve future studies in this, the great number of N was better, but we should try to eliminate more confounding variables. In this case, increase the N even more, capture exactly the baseline colony size, capture any mite counts. She treated her packages to bring them to the same level. I think that treatments can be actually impacting, it could be a confounding variable that impacts the queen quality or some of the bees. So I said no treatments. The tests need to be balanced to reduce the differential variables. I think we should extend the time frame to have a longer time period, not just over the period of a single season, but potentially over 2 or 3 years. I think we should improve the data accuracy by using a measuring grid in potentially 1-inch squares with a photographic, data collection of each of the brood frames, so it doesn't leave any, not as much to objective count. And I also think that the person that's doing the data analysis should be different from the person collecting the data, and they should not know which ones are test and control. This way it provides a blind outcome of scoring the pictures. The other thing that we probably should do is record the environmental changes across the study time, weather, forage, things like that. Because she was implying that the bananas during a dearth would be potentially helpful, and her study was during a dearth in her area. It turned out that in my area, it was the onset of a nectar flow. So maybe that caused some issues, right? So we need to calculate that as well. So we looked at the comparison of studies already. What I would like to discuss from those results. So obviously the banana fed test group recorded a whole lot less brood surfaces, which means the feeding bananas to them had a detrimental effect on the brood rearing potentially. So I'm big on looking at potential unintended consequences, especially when we're introducing foreign substances in a superorganism that has very delicate, connections and balances and homeostasis. So one of the possible issues that the bees had to deal with when they were fed bananas was the isopentyl acetate or isoamyl acetate, IPA for short, was potentially, it basically smells like alarm pheromones, right? You've heard of bananas and it smells like alarm pheromones. So what potentially could have happened is that it signaled danger and triggered this defensive behavior, or at least stress, just preoccupation on this part of the bees, and potentially agitated them, confused them, distracted them from their brood rearing duties. There might be something to that. So it might also have temporarily reduced normal foraging, colony cohesion, and brood care. So something to consider. And the studies at the bottom are some of the other studies that kind of look into this as well. Another aspect of things is that when we're not thinking about what we're putting in the hive, bananas that are fermenting and getting old into that box, that's an enclosed environment, might have fermented and evanesced some ethanols, alcohols, acids, esters, or fermentation byproducts that might have been, yeasts and molds that could have negatively interfered with the nest and the brood rearing, right? Especially because they have a very narrow gut microbiome that's very specialized and for pollen and nectar. And now we're feeding them a completely foreign substance that might interfere with their gut microbiome, and we'll look at that. So some of the potential causes of these differences might have been toxicity triggered by the banana fermenting, bacteria from spoilage, and microbacterial, microbial imbalances. The microbes in the nest might have been negatively affected, which, by the way, the brood also has a very, sensitive microbiome, so that might have decreased their appetite. This could have increased malnutrition of the brood and negatively impacted their nutrient absorption. So that might have explained why there were fewer brood. And also, maybe those smells in the hives and those compounds were interfering with pheromone communication, brood pheromone, nurse pheromones, things like that. The stress that it might have caused could have been distraction, disorientation, but also thermoregulation disruption. There might have been some of those molds and yeast. There might have been mycotoxins, as well as fungal spores that might have contaminated the bee bread, that might have been toxic to the brood. The contamination of the nest might have become actually long-lasting and chronic, and it might actually have negatively impacted their immune system, right? That also could affect their detoxification processes, right? Because it's overwhelming, potentially. And it might have altered their gut and crop pH, their hindgut and their crop, the pH with the acidity, the organic acids that were released, the CO2 as well, and the ethanol, all are acid-inducing, as well as the vitellogenesis, which, as we know, is vital for immune system regulation. Beyond that, and I will show you what I mean, it might have caused some proboscis and intestinal blockage, in particular the proventriculus, the valve, the one-way valve that's between the crop and the hindgut, osmotic stress, and narcosis. death of brood, right? So when it comes to the digestion itself, bees lack the mouth parts to chew bananas, right? And they also lack the enzymes and gut adaptations to digest banana materials, compounds, plant fibers, starches, complex fruit compounds. That's not what they're made to do. That's not what they feed off of. So their gut microbiome is also very specialized for monosaccharide and disaccharide processing, fructose, glucose, and sucrose. And they cannot metabolize cellulose, plant matters, pectin, or protein found in fruit, right? Only in pollen. So basically, they might get some nutrition from the liquid syrup that's from the fermenting fruits, but nothing else is really absorbable without causing potential harm. So this is just kind of a anatomical issue with consuming bananas. Again, they cannot chew. They absorb their food through their proboscis, and their mouth parts unfold, and so their proboscis is surrounded by harder parts, and I'm going to show you. But the galea and the labial prementum just form a straw, basically, in the food canal through which the glossa, the tongue, gathers the liquid, and then it gets sucked into through the pharyngeal pump. So the way this looks, let me see if this works on this. Basically, they do lapping and sucking. They don't chew their food. You're going to say, well, what about pollen? Aren't they chewing on that? Well, really, pollen is microscopic, microns. And the way they absorb this is through dilution of sugar nectar. It's a suspension that has been actually pre-digested in bee bread, fermented and processed, so that the pollen grains the shell can be cracked open so they can access the inside of those pollen grains, right? But it's a micron size. It's not a banana, right? Or those parts that are in the banana. So I'm going to see if that video works. So they're going to unfold their mouth parts, and that's going to basically provide a funnel for their glossa, their tongue, to go and suck in the nectar from the flower. And when they're in the hive, they can dilute it with the bee bread so that they can absorb that. Let me see if I can go to the next one. There we go. So that tongue right there is covered in hair, and they cannot chew to swallow food. But I would ask, some of the unintended consequences might potentially be that banana paste, melted, goo, might clog up their hair on their tongue, which might lead to nutrient malabsorption, right? That might actually make the nurse bees less able to not only get nutrients for themselves, but also feed the brood. I don't know, more research is needed. The one on the left is a good picture of that as well. So just some examples. So in the end, I want to remind everybody that there might be some unintended consequences that are linked to feeding foreign substances to colonies of honeybees. Beekeepers love to feed their bees all kinds of stuff, right? The banana project is one example. But they need carbs to stave off starvation. And if there's no nectar or honey, and they need some carbs, beekeepers might feed sugar syrup or pro-sweet kind of like syrups. And sometimes they like to feed protein that are actually pollen substitutes. So we have the pollen patties and the soft sugar bricks that have those pollen kind of like elements so that they can feed the brood. The problem is that when you feed some carbs and pollen through potentially a dearth, now you're keeping the colony in brood, when it otherwise probably would have taken a brood break naturally. And when you raise more brood, what else are you raising? More mites. So that might be an issue to consider as well. We really need to be mindful of potential unintended consequences. So that might lead to an increase in pests and therefore pathogen loads. It might lead to an increase in toxicity, mortality, bird rearing might burn out, and they might get a shorter lifespan. That's actually a study by Emily Nordyke that was looking at all the studies of feeding pollen substitutes to colonies of bees across the industry. And those are some of the real observations from some of those studies, right? Premature swarming might be an issue, right? When you keep them in brood, they might swarm before the nectar flow. No drones to mate with those virgin queens, that might be an issue. Or they might burn themselves out because it's still cold and they're trying to rear brood out of season. And they're going to go through all their food and starve to death trying to keep that brood warm. So starvation in early spring. And also, who knows what it does to their gut microbiome? Some considerations. So pollen patties, pollen substitutes, soft sugar bricks that contain pollen might be causing some issues. So what I always tell beekeepers is, and this is just not the results of the study, this is kind of like an example of how to keep in mind unintended consequences, is that respecting natural brood breaks it tends to be a pretty good idea, not forcing through it. So in conclusion, it appears that feeding bananas to our colonies of bees might have detrimental effects, especially when it comes to biochemical and microbiological tasks. It's not supported by bee biology or scientific evidence. So that was one of the goals I was trying to achieve. The alarm pheromone mimic can actually trigger agitation, defensive behavior, or confusion. It increases stinging behavior and disrupts normal hive cohesion, that banana. And the bees don't have any digestive adaptation to that kind of food because they have complex plant compounds, starches, and fibers that they cannot digest or even chew on. The interference with gut microbiome, brood food production, and nutritional stress in nurse bees and brood should be kept in mind. In bananas, there's no balanced protein, fatty acids, or sterol like there is in bee bread and pollen. And because of that, if they are able to absorb some of that, may displace proper foraging or pollen intake and with the actual nutrients that they need. So it dilutes nutrition, basically, makes them malnourished. The forage diversity and abundance in your area, planting for your bees is probably a better idea if you want to help your bees. Tell all your neighbors how to plant for your bees. The use of locally adapted survivor stock that's well adapted to the local cycles of weather and forage is going to help, especially if they take natural breaks during the dearth. Novel foods, always keep in mind that they can disrupt the hive's chemical communication and nutritional balance. So you might actually harm your bees. And true strengthening really comes from actual balanced nutrition that's adapted to the honeybees, pollen, natural pollen, and a natural nectar. And then also that strengthening comes from lowering the stress and using locally adapted genetics. So basically, I think that Katarina's study had major design flaws. There were some inaccuracies in her paper. And I just think that further research is needed to kind of try to examine some of those mechanisms that are causing that loss of brood surfaces when feeding bananas. So with that, I just want to mention I have some references, but I wanted to thank you and open the floor for questions. Yeah, thank you. Yes.
I'd like to ask more about the failed hive, the timing of the failures. Were they building the seed cells or anything? Can you describe a little bit more why they failed and when they failed?
Well, the why I would only be conjecturing, and it was really early into the study at the end of the first week. So it might have been due to the queens themselves. Maybe something happened when they were mating. Maybe the installation caused issues. But also maybe some of the interferences with the bananas and what kind of components were being, vaporized into the colony. Maybe some of that had an impact on them as well. I would only be guessing.
If it was, say, the September last September, would the workers be after that? Would they start constructing new bee cells?
Yes, but I still took them out of the testing because that was completely not following the same path as the other colonies at that point. So I had to remove them either way. Yes. Yes. Good question. Yes.
Can you repeat the question?
Oh, yes. So the question was like the colonies that failed and lost queens, what was it due to? And were they making queen cells? That was the question, basically. Yes, you had a question. Of various fruits or just banana juice? So has there been any studies on feeding fruit juices of all kinds? There's some look at, in terms of bananas, pumpkin puree syrup, other things that were tested, especially that was the NUPAN study. And here and there's little things, but not as a general, study that systematically looked at various fruit juices without introducing confounding variables such as honey or pollen substitute feeding, or it's kind of hard to find anything that's really pure looking at that. Hey, Lauren.
So you said you had a pretty decent nectar flow during that time. Did you have consistent consumption on the bananas? Were you throwing them out after you?
Yeah, some of that. So every week I would come in, and actually twice a week I would look if they needed more. And because the nectar flow started coming in, the question is, did I have good consumption on the bananas? And overall I did, but some of them were, when I was coming back for the next visit, they were kind of sitting there and just kind of rotting out. There were a couple instances where I saw small hive beetles in there as well, because obviously fermenting fruit is perfect attractant for small hive beetles, which could be another unintended consequence to feeding that kind of stuff to your bees. Yeah. Over there.
Just kind of a follow-up question to Lauren's question. On the control, what was the uptake on your feed compared to the banana feed?
As far as the syrup is concerned, what was the uptake?
Like, were they comparable? Or
Yeah, they were comparable, but it slowed down really fast once the nectar flow kind of kicked in, right? They don't really like taking the sugar syrup when the nectar flow comes in. But I kept feeding bananas and offered the sugar syrup nonetheless. But I noticed that it was not getting consumed as much once the nectar flow started establishing. You had a question?
Yeah, I was wondering if there was any backup information as to why packages were chosen instead of looking at overwintered established products.
That's a great question. And for the same reason, one of the studies was looking at swarms and colonies that were established. Is that it's a lot easier to compare apples to apples when you're using packages that are 3 pounds, same, grafted queens that come from the same genetics, if you're buying them from the same supplier, and they can be installed at the same time with fairly equivalent sizes, expecting fairly equivalent growth, although still there's a variation, but it's not going to be as much as an established colonies that can have all kinds of things going on. And it's really hard to compare existing colonies that are established, actually. But that's a great question.
I'm just wondering if maybe like packages that are starting out with nothing, being fed to bananas, had anything to do with the output. Like if you have an established colony and you overwinter them, you feed them bananas in the spring, if there's a difference in the outcome.
That's a great question, and I think that's something that's worth looking into. I think you're going to find that it's really hard to eliminate confounding variables in that case. So the conclusions might be subject to interpretation and might not really represent what's really actually happening. It's hard to isolate variables in that case, but yeah, more studies are needed. Yes.
So my students keep regular earthworms in our classroom, and we have to be really careful about feeding them bananas from certain sources because they kill the worms.
Right.
And so I was curious, how much did y'all control your source of the bananas?
That's a good question. I use only organic bananas, which was a little bit more expensive, but I wouldn't feed my bees anything that would potentially be contaminated by chemicals if I can avoid it. And it would help eliminating some confounding variables as well. So I used organic bananas. I know that it can be really toxic otherwise. So good question. Anybody else? Yes.
I had remembered that you had said that KD had fed her packages pollen.
Yes.
But did you, because you were in a
No. Katie didn't. That was another study.
okay,
Yeah,
okay, then that goes.
Then there goes your question. Okay, thank you. Anybody else? One last one, Bill. Hang on, I'm coming to you.
They can hear me, I should. Not necessarily on the bananas, but on the question of fermented fruit. And I could never prove it, but I'm fairly certain that I saw a bee kill once on fermented grape jelly that was thrown out. And I'm fairly certain that it killed bees.
Ah.
And it was kind of in relation to the fermentation stage of fruit that you get to in there.
Yeah, well, there's probably something to it. I mean, more studies are needed to look at that and verify that. But any fermented fruit can potentially be toxic to bees, right? It's like who knows what's in that, and it's not really good for them. Just the same way small habitats will ferment the honey, and then it's not good for them anymore. Any alcohol they might be drinking might not be really good for them either. So who knows what was wrong with that grape jelly as well. But that's a good observation. Yeah, you had one more?
And so, because you stored or started with packages,
Uh-huh.
do you believe that the IPA of the bananas actually interrupted the QMP of the queen bee?
Well, that's a very good observation, and that's one of the things that I tend to look at is anything that you put into a colony that's using pheromones for communications, for foraging, for cohesion around the queen. Feedback loops, positive, negative. That's really kind of anything that you introduce that's going to be potentially messing with those pheromones could be an issue. And that's where in my treatment for operation, and we'll talk about that later, I don't use essential oils at all because it could interfere with some of those pheromones as well. It's entirely possible that it might have had a role. And it might have had an impact on those extra queen failures in the test group. Yes, correct. Good kind of guess. Oh, yeah.
Okay, can you verify that she only fed sugar water to the control group and only bananas to the test group, or sugar water with bananas to the test group?
So the control group was only fed 1-to-1 sugar syrup. The test group was fed the same thing, 1-to-1 sugar syrup plus bananas. So does it answer the question?
I think so.
Yeah, okay.
Last one. So do you know the amount of fructose/glucose in the bananas versus the amount in the sugar water?
Let me look at that. There was a slide on that. Where's the computer? Oh, you got it. Let's look at the beginning, like one of the first slides. And it was not exactly the same, but either way, it's kind of hard to compare as you're going back to it. There's a lot of slides. Sometimes it's easier to kind of go down there. There you go. To navigate a little faster. There it is, that one right there. And then, so we've got fructose, 27%, and glucose, 28% of a banana. So that's the answer, I guess, to that question. Yeah. Anything else? Anybody else?
Other questions?
Speak now or forever hold your peace. Yeah, that's a good question. And honestly, we could construe that. So you were asking about, have we done some studies about feeding just the bananas and no sugar syrup, either in the test or the control group, meaning only one variable being the feeding of the bananas? Then you wouldn't be comparing apples to apples. Yeah, if you feed the control group sugar syrup, but not to the test group, and you only feed the test group bananas, that's a confounding variable. That's not going to work for your study. You're not going to be able to make any appropriate conclusions. Yeah. No, I mean, okay, so to a certain extent, the amount of sugar juices, that they can extract, like water with fructose or glucose suspended in that water, might give them some source of carbs that might help them build up some beeswax, which actually might explain, I don't know, some extra comb building, but not brood rearing. So potentially, but I suspect that the amount would be negligible compared to what they would find otherwise in nature or what they could extract from those bananas. Yes, yes, yes. Well, and I got news for everybody. The natural comb uses just as much beeswax as your foundation. What the foundation does is it doesn't cut the amount of beeswax needed to build the comb, but it allows it to be reused, and it also allows a one size fits all cell size, so that you discourage drone rearing and encourage more workers. Yeah. Yes.
Did you say that KD started with 12 swarms that were caught?
No, Katie did not. That was another study. I think that was the Nupan study. And no, she used all packages, same supplier.
Well, with that, Italian honeybees, which are more famously known as storing more honey,
Uh-huh.
do you think the amount of banana puree that was actually stored in the honeycombs could have signaled that stress because there was more banana in the combs versus, let's say, a Russian?
Can you repeat that? Because I
Like, do you think the breed of honeybee with this storage, being that they store more, you know,
the population grows a lot faster in
Right.
Italians. They tend to bring back a lot more nectar because of it, but they also tend to consume a whole lot more because they've got so many hungry mouths to feed. So I don't know, maybe potentially.
like the traits of storage could have played an impact on that?
Yeah, so Russians, for example, they're a lot more frugal than Italians. They're also much more resilient. They have more resistance and they're much more fit than Italians tend to be mite susceptible. But by being very frugal and very elastic, they're able to adapt really quickly to the cycles of weather and forage, and to stay small when there's a dearth. And as soon as they sense a nectar flow coming in, they just explode, like just literally explode. And then when there's a dearth coming back, they'll contract back. They're very frugal and don't use a whole lot of resources to feed their colonies. Whereas Italians, they do store a lot, but also consume a lot. And because they rear a lot of brood, they also rear a lot of mites. So, yeah. Yes. One more, last one. Is that? Yeah.
So, given that you take into a nectar flow, did you observe any spoilage or fermentation in the feed that contained the bananas?
Yeah. So 1-to-1 sugar syrup. So you're talking about the bananas or about the syrup? Both. Both of them kind of the nectar flow is associated usually with a warming of the weather as well, and because the bees are not necessarily taking as much sugar syrup, they tend to leave it in the jar a little longer. And as we all know, 1-to-1 sugar syrup, when the temperature rises, it ferments really quickly in 2-3 days. And at that point, the bees might not be interested in that. But because they're looking at the nectar flow coming in, it kind of makes up for that either way. And all the colonies were getting the same amount of nectar because they were in the exact same yard. I mean, minus variations between different colonies and potential makeup of the population and the mating of the queen. There's a lot of things that come into play, right, when they open mate. Good.
Okay, thank you.
Thank you.
Should we take a 10-minute break?
One down. Yes.